Okay, in keeping with the theme of how FDA handles food ingredients, I'm going to discuss how the agency evaluates live microbial cultures used in food under the Generally Recognized as Safe, or GRAS, notification program. GRAS is one regulatory pathway that food ingredient manufacturers can use to ensure the safe and legal use of food ingredients. Here's an overview of the points that I'll focus on. I'll define what live microbial cultures are. Then I'll discuss GRAS as a regulatory pathway for food ingredient uses. I'll discuss what's common between the GRAS pathway and the food additive pathway and what distinguishes these two regulatory pathways from each other. I'll then discuss FDA's GRAS notification program and tell you how the program works. As part of that discussion, I'll show you how live microbial cultures are among the food ingredients whose uses have been evaluated under the GRAS notification program. To demonstrate how the GRAS notification program works, I'll discuss the types of data and information that FDA evaluates when the agency receives a GRAS notice for a live microbial culture used in food. Last, we'll go through two examples to see how much you've learned about GRAS. Live microbial cultures are added to a wide range of conventional food products, including those shown here. As you heard previously, under the Federal Food, Drug, and Cosmetic Act, hereafter referred to as the Act, FDA is responsible for ensuring food ingredient safety. This includes the safety of live microbial cultures. Consider the answer to this question. Live microbial cultures are the same as probiotics. True or false? Okay, and now for the answer. For the nearly 68% of you who answered true, you are correct. For the almost 32% of you who answered false, you may also be correct, and here's why. Internationally, probiotics are defined as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. Live microbial cultures would satisfy this definition, and therefore, scientifically, live microbial cultures and probiotics have the same meaning, and A is true, or A is correct. However, while FDA has many years of experience evaluating the safety of the food uses of live microbial cultures, the agency has not adopted a legal definition for the term probiotics. Therefore, B, false, may also be correct. During this presentation, I'll continue to use the term live microbial cultures. Now let's take a look at two regulatory pathways for food ingredient uses, GRAS and food additives. We'll look at the aspects these two pathways have in common and what distinguishes them. The Act defines what GRAS substances and food additives are. According to the Act, any substance that is added to food is either a food additive or a GRAS substance. GRAS substance uses and food additive uses share common aspects. First, the chemical nature of GRAS substances and food additives is the same. For example, live microbial cultures may be used as GRAS substances or as food additives. The same holds true for other food ingredients. Under the Act, GRAS substance uses and food additive uses must be shown to be safe using adequate scientific data and information. Furthermore, the safety of GRAS substances and food additives is based on the intended use, including the use level, of the GRAS substance or food additive, and not the GRAS substance or food additive itself. Since GRAS substance uses and food additive uses must be demonstrated to be safe using adequate scientific evidence, both require what is known as the technical element. However, the Act also requires that the use of a GRAS substance meet the common knowledge element in order to satisfy the GRAS standard. The common knowledge element requires that the safety data and information used to support a manufacturer's GRAS determination be generally available to everyone and accepted by qualified scientific experts. General availability of safety data and information is usually demonstrated by publication in peer-reviewed scientific journals. General acceptance may be demonstrated by the consensus statement of a panel of scientific experts or by the opinion or recommendation of an authoritative body such as the National Academy of Sciences. General acceptance may also be demonstrated by the body of scientific literature. Substances determined to be GRAS for their intended use must meet both the technical element and the common knowledge element. The common knowledge element is not required for food additive uses, but is an additional requirement for GRAS substance uses. This diagram provides another illustration of how GRAS substance uses and food additive uses must both be proven to be safe and therefore meet the technical element, and how the common knowledge element distinguishes GRAS substance uses from food additive uses. Unless it's shown to be safe, the use of a substance can neither be a GRAS substance use, nor can it be an approved food additive use. In order for a substance to be safely and legally used in food, a manufacturer must have adequate safety data, regardless of whether the manufacturer intends to use their substance as a GRAS substance or a food additive. If a manufacturer can show that the data supporting the safety of a substance's use are generally available to everyone and generally accepted by experts, then the use is GRAS. If a manufacturer considers the data supporting the safety of a substance's use to be privately held, then the use cannot be GRAS and the use may be a food additive use. FDA's GRAS notification program is the only mechanism by which the agency reviews and provides comments in response to manufacturers' GRAS determinations. Let's now take a look at what the program is and how it works. The procedures governing FDA's GRAS notification program are laid out in the 1997 GRAS proposed rule issued by the agency. While a manufacturer's GRAS determination is required by law in the absence of a food additive petition, FDA's GRAS notification program is voluntary by law. Once a manufacturer determines that their use of a substance is GRAS, the manufacturer can then choose whether to submit a GRAS notice to FDA for their intended use of the substance. Whether a manufacturer chooses to submit a GRAS notice to FDA or not, they must still do the work to ensure that their use of their substance meets the GRAS standard before marketing their substance for use. If a manufacturer chooses to submit a GRAS notice to FDA, FDA evaluates the notice and responds to the manufacturer with a letter. FDA's goal is to respond within 180 days. The agency has worked to make the GRAS notification program transparent, and the notices we receive, as well as our responses to those notices, are posted on our website. FDA has issued the types of responses listed here, following its reviews of manufacturers' GRAS notices. These responses are: FDA has no questions, the notice does not provide a basis for GRAS, or the manufacturer has asked the agency to stop the review process. In cases where FDA has responded that the agency has no questions, the manufacturer has shown that their GRAS determination meets the GRAS standard. If a manufacturer submits a GRAS notice that does not meet the GRAS standard, either the agency issues a letter stating that the notice does not provide a basis for GRAS, or the manufacturer asks the agency to stop the review process and withdraws the GRAS notice. There are a number of reasons why manufacturers withdraw their GRAS notices, including business decisions, poor quality of a GRAS notice, or unresolved scientific or regulatory issues. Most manufacturers who withdraw their GRAS notices due to unresolved scientific or regulatory issues go on to resolve those issues and later resubmit their GRAS notices to FDA and receive no questions responses from the agency. FDA's response to a GRAS notice is not an approval or a regulation, such as that required for food additive use. FDA's response to a GRAS notice is the agency's opinion regarding the manufacturer's GRAS determination. As the maker of the GRAS determination, the manufacturer owns the safety decision for their use of a GRAS substance. FDA has operated the GRAS notification program since 1997. In that time, the agency has accepted 541 GRAS notices for review and has responded to about 500 of them. This table shows the types of substances that FDA has evaluated under the GRAS notification program. Microbials, including live microbial cultures, are among these substances. To date, FDA has received 22 GRAS notices for live microbial cultures used in food. Through its experience with evaluating the uses of various microbial food ingredients, FDA is knowledgeable about the microbes that humans encounter. Microbes are ubiquitous in the environment we live in and are found in our food, water, and air. There are typically between 100 to 100,000 colony-forming units of microbes per gram of food we eat, and about 10 to 100 colony-forming units of microbes per milliliter of water we drink. In addition to their presence in our environment, microbes have safely been used since 6000 BC to ferment a variety of foods, including those shown here. Most of the live microbial cultures that FDA evaluates under the GRAS notification program are derived from cultures that have long histories of use in food. Since humans come in contact with microbes through consumption of food and interaction with the environment, humans have evolved with microbes. The human gastrointestinal tract is home to a large and diverse microbial ecology. The human colon contains 10 to the 14th bacterial cells, which is greater than the number of human cells found in the human body. The human stomach and small intestine contain a few bacterial species, but the human gastrointestinal tract contains thousands of bacterial strains. While FDA is knowledgeable about microbes, manufacturers who submit GRAS notices for the use of live microbial cultures must still provide data and information showing that their use meets the GRAS standard. Now that I've discussed some of what FDA knows about microbes, I'll highlight the types of data and information FDA reviews in GRAS notices. This table lists the live microbial cultures whose uses have been evaluated under FDA's GRAS notification program. The uses of lactobacillus species and bifidobacterium species are the most frequently notified to the program. You may already be familiar with these as they are commonly mentioned in publications about live microbial cultures. As you can see, we've evaluated GRAS notices for the uses of single live microbial cultures as well as mixtures. And now, another poll question. The source of a live microbial culture is an important consideration in its identity. Given this, please consider where live microbial cultures used in food have been isolated from. Have they been isolated from plants, naturally fermented foods, fecal material, or all of the above? And now the answer. Live microbial cultures used in food have originally been isolated from plants, naturally fermented foods, and fecal material. So the 53% of you who answered D, all of the above, are correct. A GRAS notice for a live microbial culture should show that the culture is fully identified and characterized. Manufacturers must definitively identify their live microbial cultures using genetically based techniques. Manufacturers must also name their live microbial cultures according to the most up-to-date nomenclature available. In order to ensure the accurate identity of their live microbial cultures, manufacturers must ensure that their cultures are properly maintained and manufactured to prevent contamination with other microbes. Identity is important to safety because some microbes are unsafe. The term safe, as it refers to ingredients added to food, including GRAS substances, is defined in the U.S. Code of Federal Regulations. Safety is defined as a reasonable certainty in the minds of competent scientists that a substance is not harmful under the intended conditions of use. This definition speaks to whether the use of a substance is hazardous to human health and also accounts for the reality that it's not possible to ensure the absolute harmlessness of any substance. A GRAS notice for a live microbial culture should demonstrate the non-pathogenicity and non-toxigenicity of the culture. One important consideration for the safety of live microbial cultures is that they be non-pathogenic, meaning that they be unable to cause disease or infection in healthy individuals, unlike the foodborne pathogen E. coli 0157H7. Another important consideration for the safety of live microbial cultures is that they be non-toxigenic, meaning that they be unable to produce harmful substances at levels that are either detectable or that have been shown to cause harm under ordinary conditions of use or exposure. This is in contrast to microbes such as Clostridium botulinum, which produces the food poisoning botulinum toxin. While microbes used in food must be non-pathogenic and non-toxigenic, we know that many microbes that are considered to be safe can cause opportunistic infections in immunocompromised people. As part of their GRAS determinations, manufacturers must, on a case-by-case basis, be aware of the potential of their live microbial culture to cause opportunistic infections in immunocompromised individuals. A GRAS notice for a live microbial culture should demonstrate that the culture does not contain transmissible elements that confer resistance to clinically relevant antibiotics. In particular, the GRAS notice should include information showing that, if present, the antibiotic resistance genes would not affect therapeutic use of antibiotics in the human population. The GRAS notice should also include information showing that it's unlikely that any genes conveying resistance to clinically relevant antibiotics will be transferred to other microorganisms. Live microbial cultures are reported to have many human health benefits, including those listed here. The scientific evidence demonstrating the benefits conferred by live microbial cultures varies in strength, and how live microbial cultures confer these reported benefits is a topic of research interest. Does FDA consider reported health benefits when evaluating GRAS notices for live microbial cultures? Think about this question. When evaluating a GRAS notice for the use of a live microbial culture, FDA considers the reported health benefits conferred by the live microbial culture. True or false? Okay. For the almost 65% who answered false, you are correct. Under the Act, FDA has the authority to evaluate the safety of food ingredients, but is not given the authority to evaluate reported health benefits of food ingredients as a part of these safety evaluations. However, the studies that manufacturers use to understand reported health benefits may contain information relevant to safety. If this is the case, FDA evaluates those relevant safety data during its evaluation of a manufacturer's GRAS notice. If a manufacturer wants to make a health claim for their live microbial culture, that is done under a process that is separate from the GRAS notification program. JIFSAN's Qualified Health Claims Program evaluates health claims related to foods and food ingredients. To date, no one has successfully completed a health claim for a live microbial culture. Earlier in this presentation, I discussed how manufacturers' GRAS determinations must satisfy the technical element and the common knowledge element. I also told you how the common knowledge element is specific to GRAS. Let's look at two examples to see how much you've learned. Example number 1. Company A has generated data and information supporting the safe use of a novel live microbial culture they intend to use in food. None of the data are publicly available. Company A considers them to be confidential. Which of the following is true? A. Company A's food use of their live microbial culture may be a GRAS use. B. Company A's food use of their live microbial culture may be a food additive use. Or C. Company A's food use of their live microbial culture may either be a GRAS use or a food additive use. Over 80% of you answered B, and that is the correct answer. The data and information used to support a GRAS determination must be publicly available. They cannot be confidentially held. Given that Company A considers the data and information supporting their food use of their live microbial culture to be confidential, their use cannot be a GRAS substance use, which eliminates options A and C and makes B the correct answer. Okay, one more opportunity. Second example. Company B wants to send a submission to FDA for the company's intended use of a live microbial culture. The company has published a lot of safety data, but these do not address all of the relevant safety questions. What should Company B do? A. Submit a GRAS notice to FDA and let the agency fill in the blanks. B. Submit a food additive petition to FDA. Or C. Conduct studies to address the remaining safety questions. Excellent. Over 90% of you answered C, and the correct answer is C. Conduct studies to address remaining safety questions. Since Company B does not have adequate data to address all the safety questions regarding the use of their live microbial culture, the use may neither be a GRAS substance use or a food additive use, which eliminates options A and B. Also, the manufacturer is responsible for the safety decision. FDA does not fill in the blanks. During this presentation, we reviewed the following points. First, the uses of GRAS substances and food additives must be supported by adequate safety data in order to meet the required technical element. The uses of GRAS substances must also meet the common knowledge element. FDA has evaluated many GRAS notices for the food uses of live microbial cultures, and GRAS notices for the uses of live microbial cultures must demonstrate that the culture is thoroughly identified and characterized, non-pathogenic, non-toxigenic, and does not contain transmissible antibiotic resistance elements. FDA continues to be committed to ensuring the safety of the food ingredients we all consume. If you would like more information about how we do this through our food ingredient regulatory programs, we invite you to visit our website for more information. Thank you.
Thank you very much, Shayla, for that very informative presentation. We have time just for one question, and that question is, how would one assess the potential for live microbial cultures to cause opportunistic infection?
Most of the time when we see information about live microbial cultures that may act as opportunistic pathogens, that comes from published case reports. And I think most, if not all, of the live microbial cultures that we've evaluated may have some published case reports out there showing that they're opportunistic pathogens. And that's common, and it's important to realize that even something as benign as yeast that are used in baking can be opportunistic pathogens under the right conditions if they have access to immunocompromised people. So really anything that you might think is safe under the right conditions could be an opportunistic pathogen in someone who's immunocompromised. But again, that's usually demonstrated by discussion of published case reports.
Well, thank you very much. Very nice presentation. And we're going to move on to our next presentation momentarily.